Okay. Well, let's go ahead and get started. It's already 3 after. We did start a little bit late, so sorry to everybody who joined right on time. But thank you all for joining us. This is the COVID-19 recovery series. Today, we're going to be talking about the vaccine, which is finally here. So we're just all wondering, is this the beginning of the end? Is this really going to help us get out of this nightmare? And how does this all relate to the businesses that we're either advising or running on our own, or the businesses that maybe we're just starting to look at launching? My name is Carrie Naoni. I'm the Executive Director of TIE Oregon, so thank you all for being here. First and foremost, I want to thank our support system here at TIE. Without the folks on the screen, events like this just wouldn't be possible. So if you are looking for funding sources or a place to work or accounting services, please take a look at the folks that are on the screen. They support us, so we would hope that you would look to them for their services. All right, and again, as I mentioned, we are here to talk about the vaccine, and we are excited to have TIE Oregon member Dr. Minishki Knoll with us today to walk us through, a lot of the things that probably we're all thinking about. How is this vaccine going to work? Is it going to work? What does this mean? When can we get back to work? When can we be in person? She's also going to talk a little bit about how to dissect some of the scientific data that we're all seeing online and trying to make light of. So all kinds of great things that Dr. Minishki Knoll is going to provide to us today. So with that, I'm going to just turn it right over to her. Doctor, go ahead and share your screen and start your presentation.
Thanks, Carrie. I see the share screen, so I can do it now. Okay.
Yeah.
Can you? So it's visible, right? This is the size. It's fine.
So we can't see your presentation yet. So if you just go down and hit share screen.
I did.
Try it one more time.
Yeah. Okay.
There you go. And then just make it a little bit bigger.
Yes, it is on the side, so I'm gonna fix it.
There you go.
Oh.
You are ready.
Okay.
Hit it. Oh, wait, you know what? I forgot to mention, if you have questions, please place them in the chat box or in the Q&A box. And Nicole Maris, who's here with us also with TIE, is going to be moderating that chat box. This event is also being recorded, so if you miss anything, we will post this to our website after the event. Again, questions in the Q&A or the chat box. Thank you.
Carrie, I thought you were leaving, right?
Yeah, I'll be leaving halfway through, and okay. Nicole will take over.
Okay. So are you ready to hear about this pretty little creatures over here?
Go ahead.
So,
Yes, go ahead.
Okay. So my name is Meenakshi, and so I am the founder of the Scrutinizer. And the Scrutinizer is, as the name says, it is for all the misinformation there was. So I question the misinformation, and especially now with COVID, I don't even have to take any other example. And because whatever is happening right now, it shouldn't even have happened. So I wanted to show this for a minute. Can you hear this music? Could you guys hear the music?
Yes, we could hear it.
The reason I was doing this music is if COVID-19 could speak, this is her or its music, because how they had the genomic sequence was published in January. So basically it's AT and use and so forth. and they all relate to the amino acid sequence. So a scientist took all those amino acid sequences. He did not manipulate anything, but he put them according to the musical notes, and that's the music that came out of it. So if it could speak, that's what it is, that it is this is how it would sound. So what I was trying to show here is just that even under the electron microscope, the virus, it looks very beautiful, and then it has this voice, but it is the profile of a killer though. So these are the three topics I would like to discuss. Like I said, it's the misinformation, and then now we have a vaccine, and then, we have anti-vaxxers and so forth. But the other important thing that I realized along, or even now, is that there is too much information all over the place, and then everyone goes on Google or the newspaper, but they don't know how to decide which information is correct. And this is not just the lay people. I have taken an example today, which is from neurosurgeons, because apparently they go to HuffPost and that is their source. And lastly, then we can discuss the vaccine and how, if everything is not under lockdown, then how to realistically move forward with the virus or after the COVID. So what are the dangers of misinformation? So like Einstein always said, he's my favorite scientist, so I quote him a lot. He said, curiosity has its own reason for existing. What this means is that if we just follow, if we just agree and follow, we are not going to make any progress. And if I at least have seen anything during COVID, is it's like the people follow it. They're the loyalties, or it's the friends, or it's the opinion. And that has done a lot of harm in how COVID was handled, especially in the United States. So if we do not ask the question why, then we don't need anything, and so we will not be moving forward. And then if we are going to ask the question why, then we have to be brave to have the answer, whatever answer it brings, because science is about facts. It's not about the opinion. This is what has gone wrong in all this COVID, I would say the COVID saga, because it's like everyone has an opinion and no one is following the science, and the scientist is not really doing anything. But the non-scientists, they have an opinion, and they so it's like a mess. So that's the reason I wanted to discuss the dangers of misinformation. Um, so so why is it harmful? Some of the things that I just discussed it, but so if there is there is distorted truth, or there is so much information, like even I cannot decipher it because there is too much. And sometimes it seems like people will say things very confidently, even if they don't know. So normally here I take an example of, for example, the anti-vaxxers or the GMOs and so forth, but there is a big, what I was afraid all this time regarding the anti-vaxxers was that if measles come back, which they have, the one good thing about measles virus is, and now you know a lot about viruses, so I think you would appreciate the example of measles here, because measles is also an RNA virus, just like COVID-19 is. But measles virus is very stable. So that's how we got lucky, that at least when that outbreak had happened, and measles had killed more people than COVID. And I don't want to hear from anyone when they say, well, measles killed more people, so COVID is doing it. Oh, what's the big deal? Or they'll say, well, Spanish flu. I mean, Spanish flu was in 1918. We are in 21st century. We knew what to do, but we didn't. You see, this is what the misinformation or the distorted truth does, because everyone just made a truth what they wanted to believe. when the doors were closing, most of the people told me, I'm not worried about catching the flu. I'm worried, can I come back home? I have a good immune system, so I am not going to get ill. So if anyone of you know anything about 1918 flu, who was dying? Who was the target population in 1918? It was the youngsters from 20 or up to 30, because they had a good immune system. So that went in overdrive because of that flu. Old people were fine because they did not have that much of an immune system. And so they were fine, but the youngsters were the ones that we lost. So the point I'm trying to make is that if we interact something new, we cannot just sit there and say, this is safe because I don't know. I mean, nobody says this, but I have actually, a lot of teachers even came to me and they said, we don't know why, but initially we did think it's just the flu. It's not going to do anything. And then this is what happened. Otherwise, as somebody who has studied epidemiology all their life, I would say we shouldn't even have faced the COVID. So from all this rant of mine, the point I'm trying to make is that we need to follow the facts, because and science there is no gray area, so it's black and white. So this means, and there is no room for emotions. And like I always say, I'm a scientist, emotions are alien to me. So we're going to follow the facts, not opinions, belief, and this is the only time I've seen that there is deadly virus out there and everyone just talks about the politics or there's a blame game. So, so the facts, what are the facts that we are facing in novel virus? Yes, we know the coronaviruses. There was SARS, there's MERS. That is a big family, which is called a coronavirus family. And all these other viruses, they come in that family. So similarly to humans, every human would be different in one family. So if a virus is new, we don't know what it's capable of. I mean, this virus showed us. However, it was hard to handle it because normally, because it falls in the same category as the influenza or the flu or the respiratory viruses. So normally all those viruses would have same kind of symptoms in all the patients. This virus it causes neurological symptoms in some, it causes GI symptoms in some, there is upper respiratory, there is lower, it's like everywhere. So now to handle that and then not follow science, it is a big mess that has happened. Because the reason I say this is that CDC, the American CDC, had 83% preparedness, and this is the highest in the world. And what does CDC do? CDC is their job. They are like army. Army sits and looks for the enemy that they can see, I hope. But CDC is the one that are looking at the bacteria and the viruses. So every flu season, they go out, they collect the data, and then they sit and interpret the data to see which strain of flu is going to be coming next year. And that's how a flu vaccine is made. Because flu vaccine is also an RNA virus, just like COVID is. But the flu virus is highly unstable because every time it replicates, there are mistakes in it. And normally it's the DNA to RNA to protein, but because this virus is RNA, so every time that RNA replicates, it's going to make mistakes. And mistake meaning then the vaccine we make, it's not that effective. And for COVID, so for COVID, this is what CDC knew that that COVID is coming. I mean, we got lucky because normally pandemic doesn't come with a warning. So pandemic, in this case, China went to WHO. So we even knew that a deadly virus it would be here because it's the travel. But from there, we didn't make any mandates. I mean, there was nothing. And then we didn't even have a test and so forth. But if you ask me, it was written all in our 101 book. It's Epidemiology 101. There are only 3 things you do, and we would have just passed it. It would not have come inside the country. But now it is here. So now it is here. So I just wanted to request that everyone just take a moment and think about the facts before just getting carried away with New York Times or HuffPost or the political parties and whatnot. So, oh, I messed up my slide. Okay, so this is what I was saying about CDC is, and even now the CDC has not made any mandates, but I don't know, it's just too late for lockdowns or what they can do. But if we follow the 3 mandates that were given to us, so regardless of anything, you would be fine. Because what happens about the virus or any bacteria is, it's not that it goes in our mouth and immediately it starts reproducing. No, there is a window, like almost 2 or 3 hours, because anything that goes in our mouth or nose or wherever, so it has to attach to the mucous membrane. And that is where it takes time. So this is where the whole thing comes, as if you were outside, you come home, you wash your hands with soap and water. So soap is the key part here, because what is the virus? Virus is, it's nothing. There is an RNA little twig inside, and outside there is a protein coat, and there is a lipid layer, the fat layer. So that's why soap comes in the picture, that soap is going to disrupt that lipid. So then it is not, it is going to, RNA alone is not going to be alive. Second is the same thing with the throat. You can do, I always do it, that you take warm water and you put salt in it, because the goal is that you do not let a bacteria or virus sit on your mucous membrane. So if you come home and you can do the gargle, you washed your hands, and of course the proper disposal of the mask is very important, because we might think there was nobody who had COVID, but we don't know, because there are asymptomatic people also.
Dr. Knoll, we do have a question about the RNA. Are RNA viruses harder than others to create a good vaccine for?
No, that is easier to create. Meaning the time that it takes is very little, like a month you can make a lot of virus. And the reason I said it's easier to make, because by now, I mean, we make RNA every day. I made RNA every day in the lab. So you make an expression vector, So for this one, Moderna was actually making it 2 years ago because first they were doing for Zika virus, and then they were doing for SARS when it came in 2002, and they even have mouse data. They actually made animal model to study it, because that this SARS, MERS, and even COVID-19, they do not attach to the mouse receptor. So it's the same receptor, the ACE2, in humans and in those animals, but this does not recognize the mouse. So Moderna even had, or they still have it, these mice they made, they have human ACE receptor expressing in them. So that this is how we do the animal model studies. And you just don't start taking humans for everything before knowing what can happen. So they already had the RNA expression vector made in February, because January we had the genomic sequence. Then we had this music also. So Moderna already had made that RNA. And so now they only had to, so this means they had already done the basics. because with RNA, what happens is it's very labile. So if you put RNA in the body, it just gets disintegrated. It will not do anything that you're asking it to do. And this is an RNA. So there's a distinction. There are 3 RNAs that we have. So this is an mRNA, which means it's a messenger RNA. So messenger RNA, it acts exactly like a mailman. You tell it, okay, go here, give them this. So this is what mRNA, this is what the 2 people have done, Moderna and Pfizer, that that they have put and it's a synthetic piece of RNA, and it is not going to make the complete uh, spike protein. So, but it has information in it that as soon as it goes in our muscle, it is gonna, our muscle cells are gonna think that what they are given orders are to make this protein. So they are gonna start making that protein. And so that protein is just gonna be in the circulation where our the T cells and B cells, they would be expressed, or they would come out wherever they live. So they would try, with neutralizing this protein, because right now it's a foreign protein. But the first response is always a weak response. That's why we need a vaccine. So we did this. Now the body will have a memory, so that the second time, if the real virus we do encounter, so that would be a strong, hopefully, because until we have done it and looked for so many, this data has been for 6 months, what Moderna has. But that is not enough time to say anything. But what they did see is it reduces the symptoms. So it's acting like more like a influenza virus vaccine, that the people still got who got COVID after that, it would reduce their symptoms. Now, from this data, we cannot say whether it is gonna protect us or it's not gonna cause the disease. Because when they made their design for the clinical trial, the goal was not to see prevention of the disease. The goal was to see, does it protect us? So because of that, they had 30,000 subjects they've done the study in. But because of that, they did not screen all 30,000 of them every week like they should. They only screened when the subject came and said they have symptoms. So that's how they have 94% efficiency. I did look at the numbers. So that's why right now, even if you take the vaccine, we will still have to have these mandates of social distancing and a mask, because if the vaccine is only reducing the symptoms, or it's in a way, it's a false sense of healthiness, because we all could have been infected by the virus, but we would not know it because the vaccine is not going to let us feel sick. However, we would be transmitting the disease. And so there now, it's tricky in other sense that normally the whole population should be vaccinated, but this is an RNA vaccine. And anytime you put genetic material in the body, we do not know. There are some concerns always, but the concern is not as it would be with a DNA vaccine. Like AstraZeneca's vaccine is different. AstraZeneca, it has made a DNA vaccine, and they have used an adenovirus. Adenovirus, it's not that adenovirus is going to do anything, because it has been, you take its infectious particles out, and so it is just a vector, so that you can send it inside the body. And I think I don't remember, I think they said 80% efficiency they have, but they are still doing their studies. However, I would be a little bit wary of taking a DNA vaccine, because DNA will integrate in our body. RNA doesn't. So mRNA is you send it in, even though this RNA is self-activating RNA, so that we can have a strong immune response. We don't know if we would or there was yet, but that is always the hope. And these are all the normal things that happen when you make a vaccine. So with the DNA vaccine, the DNA, the way DNA goes is it has to go inside the cell into the nucleus. It has to integrate, and then it is going to divide and replicate and so forth. But for RNA, it only goes up to the cytoplasm. It does not go to the nucleus. And then also because it's a part of the spike protein, so our cells would start making parts of the spike protein because they are parts of it. So there would not be a full spike protein made. Just those parts would be foreign parts for our bodies. So our defenses should come and neutralize those. But then, like I said, once we put RNA in the body, we cannot control it until and unless we were using some other technology like CRISPR. But the only concern I would have is, one, we should have seen it in the clinical trial, because RNA is something that always causes an inflammatory response. This is its nature. So, but that, if we can control it with antihistamines and so forth, so it should be fine. The second thing with an RNA happens is no matter how much we purified it, because it's gonna go replicating because it's a self-activating RNA. So it would make a double-stranded RNA. It's possible it won't make it, and the chunks would. So there are all these possibilities. So there are these theoretical questions that we always have to keep in mind. But looking at the data so far, but then it is not that much time to study and say we have studied a genetic vaccine which was never approved earlier, so it has no side effects, and it may not have any side effects. I'm not saying that. I just want to, because this is what I do, I have to give the entire information to the people. It's the pro and the cons, because just because we don't want to hear that something might not be good doesn't mean we should take it and then get sick or something. But I did not say that RNA vaccine is not good. I was just saying that I would be a little bit concerned if I have to take a DNA vaccine. And but RNA, these are the only two. Well, there is a third concern with it, but we don't know. We have never even seen it in the mouse once, that it would. So it's the same RNA that should boost our immune system and protect us from the virus. It's the same RNA. It can go in the opposite direction and it can suppress our immune system. But I have seen all the mouse studies that Moderna had done. I think there only one study had shown something had happened to the mice along those lines. And all what they are showing so far, what they are showing is that out of their 15,000 people, so 30,000 were the subjects, they divided them half and half. Okay, half was placebo and half was with the vaccine, but it's blinded study, so nobody knows who's who. And then they said that there were a total of 95 people, they got COVID. And then when they looked at that, so there were 5 from their vaccine arm and 90 were from their placebo arm. And they are also saying that the 5 from the vaccine arm, they did not have severe symptoms. They just had like a fever, or there was soreness, or so swelling at the injection site and so forth. But the 90, they were sick, but then they recovered. So I think this is all there we know about. I mean, the data looks pretty good for RNA vaccine. However, I still think that the phase 3, it should be that who wants to take the vaccine, they should, and then they should study their phase 3 people for a year or so to see that it is completely safe and it doesn't do any funky stuff. So having said that, the question still comes is, now I gave you all that information, and you can look at, actually Moderna, if you go to the Moderna's website or something, they have entire protocol and everything there. So instead of reading in, New York Times is actually reporting really, I don't know what word I should use, but it is just not right, whatever they have been writing, because this is how I had this discussion with the rest of the, they're the clinicians, they're surgeons and even family practitioners. And there is so much confusion after reading their articles. So I would say for the vaccine, and it is even published in NEJM, it's a really good journal. So just read the abstract. You don't need to go into details of science. The abstract is just, it tells you how many people there were and what happened. So I was going to say that at least, you know, that think critically, look at all the facts, have an open mind, listen to everyone, but then boil down the information from there, because I'm pretty sure if all of us try, we can tell which are the good facts or not. But if anyone wants to confirm it or something, I always reply, I always answer. So you can always ask me. And this one is about think critically. What I mean is, people go to Google. I was surprised that these clinicians sent me as many, they write like, I don't know, 1,000-word essays for why they should not wear a mask, why they should not be social distancing and so forth. But the references they provided to me were New York Times, HuffPost, Google. There was no scientific reference in there. And that's what I also wanted to discuss. But before that, this is the way the critical thinking or a science-based evidence or the science-based information comes in, not evidence-based. I wanted to distinguish that, because evidence-based information is, evidence is that you saw it once. Okay, so now we had a vaccine trial, right? They could have just taken 10 people and say, well, nothing happened to them and the vaccine is just great. That is evidence. It's not a science-based information. Science-based information would be where you take entire data, where you have to take more than one occurrence. That's why when we do an experiment, it has to be at least an average of 3 different times with 3 different models. So here, I'm trying to show that why you need to think critically. This is because, so here they're showing the 2 kids, and they got the vaccine. So they were autistic already. So the 2 autistic kids, they got the vaccine. So now evidence, if you're looking at evidence, it's easily, you can say that the vaccine made them autistic. The autistic children here, they're playing with the stuffed toys. Evidence-based study would say the stuffed toy gave these kids autism. So they became autistic because they were playing with these. So the last thing shows, here is a cat, it has 4 legs. And now you have horses, they have 4 legs too. If you're not following the science-based information, evidence dictates, you can call, and anyone can call this cat a horse, and you cannot stop them because, all of them have 4 legs. So according to evidence, they should be called whatever they want to be, a horse or a cat. So these are so it's not always that simple for science to make it, or to explain a mandate. For example, now for this pandemic, everyone is, or most of the people are very upset, and they would, especially over the masks. if the clinicians are upset, I would say they have transferred this to their patients. And so the patients would be confused because they'd be like, well, he's a neurosurgeon. He said we don't need masks. And so, you know, and they have gone to the extreme of saying this is tyranny. We are slaves. And I'm like, do you want to live or, you know? So it's not, they're looking at one dimensional thinking that, okay, you know, I'm not at risk. Old people are dying. So why should I be sitting inside the house? But this is not how the virus thinks. Virus doesn't think, first of all. But even if nobody's at risk, as long as the virus meets a new person it can infect, it is alive. You see why virus does not infect entire population at the same time. It will never do that because that would be committing suicide. Because if the entire population got sick, our virus will not find a new host, then it will have to go back into its dormant form. Virus doesn't die actually. It's very hard to kill a virus or a fungus. So the virus is going to do is, if it does not, so the whole idea of lockdown, first of all, the idea of lockdown was that you just stop the movement. Stopping the movement, but that was supposed to be done in the beginning when we heard it, so that any of the people, they came in and out of the country or in or out of the state, we can trace them, we can watch them and curb the virus over there. Because if people are really distant places, the virus is not going to find the new host as it wants to. And so then it just goes into dormancy. So looking at COVID-19, even if it goes in dormancy now with the vaccine and so forth, but it's possible it would come every year, because that's it is an influenza virus, and this is the behavior it's showing. And the second thing, we don't know, because so far COVID-19 it hasn't mutated much. It did make this scary mutation that it has now, but it hasn't mutated like any other virus has or the influenza virus does. But it's also possible it didn't need to because we were not protecting ourselves from it. So it just had all these hosts. And so it, you know, it's like, oh, all that food. But now when we start giving, when the vaccine comes out or everyone is getting vaccinated and the vaccine is working, let's say, then we don't know how this virus would act, because there would be a selective pressure now. So it's possible it would mutate. But that's the reason RNA vaccine is made. We didn't, that for this one, they didn't try making the vaccine like we always do. Because in the older vaccines, measles, mumps, rubella, I mean, we have them from century. So there what you do is either you take the inactivated real virus. So it's not causing a disease, but it's causing some effect that our body knows, and we have our antibodies built up and the memory cells. And as you can see, vaccine does work wonders. We eradicated those deadly diseases. There was smallpox, there was measles, there was mumps, leprosy. I mean, I can keep going on, but that's how we lived here in this. I guess that's why nobody really knew what a pandemic would be, or if they don't take precautions from a deadly virus, what it could do. So, so if the virus, if the vaccine is working, and we don't know how the virus would act, but mostly the RNA is a spike protein. So even if it mutates, it doesn't mutate in the spike protein, because if it mutates in the spike protein, it's doing us a favor. It's already killing itself. It cannot attach to our receptors. So those are the things they remain to be seen, what happens with the vaccine. However, all I know for now is, okay, it reduced the symptoms, but that doesn't mean that you can, there's another thing with this virus is even within the same family, it acts differently. So in different people, we can say it's they're different races, they're different people. So the virus acts differently. But it also acts differently within the same family because we have genes and then we have those forms of genes which are called alleles. So this virus goes up to the alleles that there have been families who were living, for example, the parents are living and there are two of the adult children there, and they were sick. They got COVID. They were sick. They had all the symptoms, but they did not have any antibodies. But they had T cells. You see, so they developed half of the immune system against the virus, but that immune system that needs to be studied. That whether it's possible that the T cells alone were robust because they have a memory also. But the people living with these two people, they had nothing. I mean, even if they were exposed, even if they had mild symptoms, but you can do antibody tests or anything on them. You can't because there are no antibodies. And one of the reasons I mentioned this antibody test is because I was listening to one of the previous a webinar where they were saying they would make this card where you can do the test at home, and then if you're traveling and this card says, well, you're not exposed to COVID or you don't have COVID. But right now it's very tricky to catch this test for the virus. So I don't think you can do it at home. And an antibody test antibody test is never the first line of test because antibodies take time to be formed. Because first there would be the infection and then they're going to take a couple of weeks perhaps, or maybe at least 7 days. It all depends on the virus. So if you don't have antibodies, doesn't mean you're not ill. Because the first line of screening or the testing is always with the PCR. And that also you do that, it's the capsid of the virus, because that tells you it's an active infection. Because even if you have antibodies, you don't know, maybe you had an infection 6 months ago, right? So that is never a screening thing. So I hope they're not making this card and saying, okay, we can do the antibody test and then nobody's ill, because a lot of people are not even developing the antibodies against this virus.
Dr. Knoll, we do have another question that's interesting on the antibodies. Do you support experience-based evidence when hundreds of thousands of individuals, along with physiologically plausibility, similar to the loss of taste and smell with COVID?
Mm-hmm. So that is, if you just gave me a number like 100,000, even if they were 100 or 200, so it's not just an experience. This is data. So if there are so many people and they are reporting, for example, this loss of taste and smell, that becomes the data. I mean, you're not saying, oh, well, there were 3 people, they reported it, 5 people. So this is how the data is. It's experience, because they are the only ones who can tell us that, okay, I've lost the sense of smell or taste. So is that what you were asking me?
I believe so. So yeah, this was a question from a participant in terms of the experience-based evidence.
Yeah, this is science-based evidence. So okay, if that's the one person, but they still need to go to the doctor, because normally you lose the sense of taste or smell after you had a flu, not before that. So in case of COVID, after we got, and I have my article, I had written this article also when we saw it from the frontline workers, that they were all healthy, but they just didn't have, all of a sudden, they didn't have a sense of smell or taste and so forth. So you never lose the that sense before you are ill. You always lose it, it's after the virus or you had your stuffed nose and so forth. So this was also a very bizarre symptom of this virus. And yes, it is a science-based then if there are so many people and they all had this symptom. So yes, that's not an evidence. Evidence would have been there were 3 people and they just went from the entire population. If there are like 5 people, that is just an anecdote. So we can't be sure. However, it is still a new symptom. So if I don't know how the doctor would look at it, but a scientist would make a note of it at least so that we can go back and look at it.
That makes sense. Thank you.
And so this is the, do I have time or because, you know?
Yes, yes, you do.
So this is the last one because I already discussed the vaccine. And so otherwise that was the last, but this is what I wanted to say about the science-based medicine and the data. That what I see is the reason misinformation spreads is, first of all, people are attached to the outcome because what they want to be true. So everyone gets the newspaper or the news or even a journal article, but they would take one study that comes out and they would say, this is what it is. So we can never take one study and say that this is, you know, this is the news. Because, so what we need to do is we need to take, even for a non-scientist, lately, because of, you know, the way everyone is reporting data and everyone is just saying things that honestly, when I have to reply to anyone, I had to actually study my immunology Bible again. Because some people say things with such confidence when I know it's wrong, but I have to go confirm it. So for everyone who is not a scientist, the best would be, so these are the guidelines I give you, and then you can confirm with me or with other scientists or somebody if you want to, because I am not saying you should be able to decipher a scientific article, but then don't believe the one article either. You see, if you cannot look at the 5 articles and then decide, then don't take one and say what's the truth. And the way it's done is, let's say I'm looking to see how I can develop any drugs against COVID. So I am gonna collect all the studies as long as I can find them. They would be 100 years, 50 years, you know. So all of them. Some of them would be really crappy studies, and but we have to collect all of them. This is the first thing. Secondly, there would be negative data. We need those also, because otherwise some people would do is, this says and nothing happened, we're not going to look at it. This one is not what I like, so I'm not going to take it. So that's not how, because you want to find the truth. You want to do what's best for you and your family. So we should not be acting like a pigeon. We did the pigeon thing already with the pandemic, that, okay, if I don't look at the flu doesn't exist. So the first thing would be, take more than one study, and negative and positive both. Don't just go for one positive, because then it's biased. Then you don't know the truth. And the science is only about the truth. So from there, I mean, I don't know, but then I think that a lot of people can look at the statistical analysis and then draw the conclusion. But if you don't want to do any of that, but you cannot take one study, take a few of those. Like they should be separated out. Like there is a negative study, there is a bad study, because bad meaning it's not a good design and they have not addressed everything. So there's a lot of factors that go in before you say what the conclusion is. So here, I took the example from, this is a neurosurgeon, like I was saying, then he sent me a number of articles, but they were mostly newspaper, because he is really bent on saying he said, wearing a mask and trying to protect us from COVID is like keeping the mosquitoes out of a barbed wire. So I wanted to tell him how to look at the data, because he did send me, and I appreciate if any clinician is trying to, look at the literature, because that's not what they're trained for, or not, they don't have interest either. So when he sent me, and so this is a good example I wanted you to show you, because, okay, so here it is. Meenakshi Noel, this is his, I just took one sentence because he writes very, very long comments. He said, Meenakshi, we do not wear surgical masks to prevent viral transmission. And these are his words. However, a typical loose-fitting surgical mask or a cloth mask is not made or intended to be used for, so it's redundant, because he cannot understand that it's not, the mask is not made for a dust or the virus or the bacteria or an elephant. It is made, there are pore sizes. So the masks and the cloth, I mean, we all know, so there are pores in them. And so according to the pores, whatever is smaller, whatever is bigger than that pore, it would be sticking out. I agree that the surgical mask and the cloth mask that we wear on everyday basis now for pandemic, it has a larger pore size than our virus. Virus is 0.12 micrometers. And so only N95 is the one that would provide 95% efficiency. That's why its name is N95. But N95 is like a clinician needs to wear it because they come in contact with the patients. Now, if the rest of us, we are doing social distancing and then we only run out to do errands, we should be fine. We do not need an N95. I wanted to mention that because even the pore size are larger for the surgical mask, but it has a really complex network. And I have an article on my website if anyone would be interested. And I only write for lay people. I don't write in science. I write science in English because I want everyone to think that, okay, we are part of our decision making and we have made a good decision for us because we understood what's happening. Because for the longest time, I think it's a nurse and she doesn't even understand why we have a lockdown. She thinks we are under lockdown, so the hospital is not full. That's not the reason. Lockdown is so when we don't meet other people, if there's a virus on that person, then the virus can lose foothold. We may not even need the vaccine. It might even go into dormancy if we can do this. And I know it has been a long time, but that's because we didn't do science all this time. We wanted to blame or whatever we were doing. I am always called a leftist or something. And so I told this surgeon, I'm like, okay, so that's why you don't understand science. Who thinks with political mind and we're trying to combat, a virus. So coming back to this is the journal he sent me from where he made this expert opinion that mask, because now he's saying the mask should not be worn in the OR either when he's doing a surgery. So the guidelines that I wanted to say is, and this is what he's written here, does evidence-based medicine. If you look at, so this is the name of the journal here, J Journal Ayub Med College something. And then you have to always look at the year. This is 2009. We are in 2020. There's no way you should look at a 2009 reference and say, they said that in 1980, the mask did not work. This is a review article, and a review article usually is very good because review means from this date they publish, they have gone back to each and every publication, and then they did a meta-analysis, and then they have published it. But this one, first I'm telling you about the year. So 2009, no, or 2000, at least say 2018. We are in 2020. Then secondly, the problem with this journal here is this is a local journal. If you want a good study, you want to know the truth, it has to be an international journal. This is where the competition is, because if they would have sent this study into an international journal, it would have never been published, you see, because the standards are high. And the third thing that everyone can do is whenever you have a journal article, the name of the journal you put in Google and look for its impact factor. Impact factor means how good the journal is. What standards are those? Because higher the standards, they cannot have a crappy study in. So this journal has an impact factor of 0.25. And just to give you an idea, for example, the journal Nature impact factor is 45. I've never even met a journal that had a impact factor of 1, let alone 0.25. However, I still read this very thoroughly, but so then I looked at the experimental design. I looked at their because it is a systematic review they have done. So in this, you look at the clinical trials, and then you see how they chose what they chose and what the experimental design was. That's not what I'm asking you to do, but I did this. For you, you should be able to get away with a year, at least, within the past 2 years, but a good impact factor journal, and then you read the abstract and read the discussion. This is all you need to know, and you would have a good idea of what you're looking at, rather than, because if you're going to choose a journal, because this surgeon has done this, every reference he sends me is the first sentence he finds which he likes, and then he sends it to me that, look, this says this. No, he is attached to his opinion. So, but the journal is not saying that. This journal said, and so it's published in 2009. So these authors are saying that in 1980, the mask they wore in the OR, rather it increased the infection. First of all, 1980, again, it's 2020. And secondly, from this kind of systematic study, there's no way they can make that conclusion. So in nutshell, this is not a study you take and then you make a mandate from it, or you get attached to it that this is what this said. So then you have to, and now there are plethora of, there are really, really good articles of 2020. They are showing, because everyone wants to, if you ask me as an epidemiologist and an infectious disease person, I think we're wasting time when we now in 2020 say, why do we need a mask? Why do we need social distancing? Show me a controlled study. This data is from 100 years ago when smallpox happened. So we have learned from that time. So to question the body hygiene, the environmental hygiene, the hand washing, because people are questioning, well, I haven't seen a study, I haven't seen the proof that hands should be washed. So what are you going to teach your children? That it's okay, you play in the dirt? Some things we learned as we evolved. So there is another study that he sent, which WHO has supported, and that's a study from China, but it is a really bad study because their data is all over the place. But more than that, apparently they were not counting that anyone reads the references. I read each and every reference. And they have put references in there without reading. So they haven't even done their due diligence to look at all the literature that was present until they published the paper. So 2020 January. And so in that review article, their last year, it ends at at 2001. So between 2001 and 2020, there are a number of great articles. they should have taken that and then made a mandate. But the reason this kind of study and their conclusion is very dangerous is because now it circulates in the people, right? So they are writing in there, the masks don't show any promise. The hand washing did not affect their outcome, meaning people who wash hands and they don't wash hands, they both had the same kind of disease or they did not have a disease. But they go to the extreme of saying they did not find they did not find an evidence that the sneeze etiquettes uh, that they would prevent the spread of virus or even flu. I mean, you don't want to catch cold. I don't want to catch cold. So they have written all these things. And then I discussed it with high school students because I discussed a lot of things with them and I tutor them. But I had thought that these students would actually think about it and say, oh, but they were very happy to hear it. And they said, well, if CDC says we don't want to wash hands, we're not going to wash hands. So you see how misinformation spreads. A bad information, it goes to anyone. These were high school students. It's not their fault, but even the adults. And then they think, oh, CDC said this and it has to be true. So that's the reason I really try, and as many people I can reach, that number one, I want everyone, even if you trust someone, my daughter, she trusts me, but when it comes to reading something, the fine print, she reads the fine print. So it's just that all of a sudden I saw within the past, I don't know, decade or so, it's more like everyone just follows their, the friends or the people. So I thought that, but when every time I ask them questions, then I've seen, then the brain's juices start flowing and they like it. So that way you would make good informed decisions because the other people can be wrong also. So why make a wrong decision thinking the other person knows better? So confirm it. So that was the whole idea of mine for the scrutinizer and the whole idea of all this science-based medicine, that I tell everyone to have an open mind, but not that open that the brain falls out, and but one thing I forgot to say, which I always say in the beginning, I do not want anyone to trust me or take my words for a fact. No. But I want you to keep an open mind and I want you to listen to my facts. And then I want you to go and think about our other facts and everything else, then make a decision. But if you need to ask me again, that is a different thing. I'm here. So the point that I'm making is I don't ask somebody to trust me just because I have creds. No, I want everyone to think, be an independent thinker, not, we want herd immunity, but we don't want to become a herd. So that's all I wanted to say. Thanks.
Excellent. Thank you so much. Do we have, if anybody has any questions, you could put them in the chat. I thought that was very informative. Learned a lot about how the vaccine actually works in the body, which is good to know, and the difference in the different types of vaccines, and it's good information to know as we move forward.
Yeah, I thought that I didn't want to go into, if it was any other day, that to show, or actually I wanted to draw those little squigglies that how the vaccine is going where, but to say about the immune system, how everyone uses the words, but they don't really understand it. And the explanations I've given is because I get asked all these questions all the time. So then I realized because sometimes I just don't realize that everyone doesn't know, a lot of immunology or something. But so I tried making it according to what people have asked. And I saw that they don't get this answer, that everyone has to have questions about this. Is a new vaccine. We've never had a RNA or a DNA vaccine. So they should be able to have like any number of questions. They should have any number of answers to their questions.
Right. We just have a few minutes, but can you discuss serial infections or reinfection in the same person and how prevalent this might be?
Oh, for COVID-19. Because normally if we had even for influenza in the same season, usually if you had a flu, you don't really get the flu again. If we had a bacterial infection, usually you don't get it because our body would form antibodies and a memory, and then they fight. But this was another unique feature of this virus, that people who did get infected and they were ill and then they recovered. So there is not a percentage because it's happening, but it's happening all over the world actually. Because I do talk to other colleagues in Europe and all. So there is not a percentage, but I cannot say, but that it's 50% or 40%, but it's not more than that. But it is less than that. But a lot of people are getting reinfected with the same virus, because this virus hasn't mutated, like I said. So it is the same virus. This means we did not develop any immunity against it. Even if antibodies, now they said they were there until 6 months, let's say. It started from 2.5 months. So they'd stay between 2.5 to 6 for this virus. And so after 6 months, they are gone. Gone is fine, but because there should be a memory of those antibodies in our system. But if we cannot detect those, that means it wasn't a robust response we had. If we don't have a robust response, then we can get ill with it again.
Okay, thank you. All right, I think that was all the questions. So I'm just going to close us out here. Okay, that was really great information. Thank you for sharing that.
Anytime, because I want people to have the information and feel that they have a say in their decisions.
Yes. Well, thank you, Dr. Knoll, for joining us today. And then I just want to let everyone know that some upcoming events, we have Pitch Club, which is tomorrow, December 15th at 5:30 p.m. And this is the final tie event of 2020. So hope you can attend. The link to sign up is on the website. And then again, this couldn't be made possible without the support of our supporters here for TIE Oregon. And we just want to again thank Dr. Knoll for joining us today, and hope everyone has a good rest of the year, and we will see you again in 2021.
Thank you, Nicole. Thanks, everyone.
Thank you.